Our next speaker is Professor Jacqui Matthews, who is Chief Technology Officer at Roslin Technologies, and we're moving from feed to the potential of IB in cellular agriculture. Jacqui leads on strategic partnerships and R&D programs at Roslin. She has extensive experience in R&D in funded projects and is very widely published. And in 2017, Jacqui was awarded the RCVS Fellowship, and so we're truly delighted to hear you speak this morning. And Jacqui, if I can, I'll pass directly to you.
Thanks very much, Jim. I'm delighted to be here, and thank you to the IBioIC for the invitation to introduce a little bit of background about Roslin Technologies, and then a bit about what we're doing in the cellular agriculture space. Next slide, please. So Roslin Technologies is a biotech company that is working to transform science innovations to provide solutions to global challenges, particularly in the ag tech area. We're located at the Easterbush site of the University of Edinburgh in the Midlothian Science Zone. And as a company, we are a joint venture between Edinburgh University, who's currently 50% of our shareholders, and two investment partners, Trust International via the British Innovation Fund, and JBI Equity. Part of our unique model is that we have the first right of refusal on all unencumbered IP that comes out of the Roslin Institute and the vet school at the University of Edinburgh, which provides us with a platform to many different projects, particularly in the space of ag tech and animal health. Next slide, please. Our main focus is in the area of the protein gap, and with the global human population estimated to expand to over 9 billion in the next 30 years, this will create massive pressure on protein production worldwide. With respect to this, meat output itself needs to double, with dairy and fish production to almost triple in this timescale, which is putting a huge pressure both on the production pipeline as well as the downstream environmental impact and food security concerns. Next slide, please. With this in mind, our focus is to address the protein gap in three main themes. Firstly, to try and increase agricultural efficiency by delivering state-of-the-art breeding programs to evaluate and improve farmed animals. And we already have revenue-generating projects in pig breeding with respect to this focus. We also aim to support the food chain by tackling infectious diseases in livestock by developing tools to improve animal health and efficiency, biological efficiency, and that includes the development of both vaccines, vaccine delivery systems, and diagnostics for infectious disease. Finally, and the focus of this morning's quick talk is we're developing alternative protein solutions to try and bridge the gap in protein requirements over the next 20 to 30 years. Next slide, please. Our two projects in this area currently are a black soldier fly program, which I'm not going to focus on today, but you can find out more about this project either through directly requesting from me or visiting our website at roslintech.com, and the derivation of animal cell lines and resources to support the cellular agriculture sector. Next slide, please. The cellular agriculture program that we're developing is part of our overall stem cell platform, and we have a unique IP and proposition in the derivation of induced pluripotent stem cells from animals, including so far from canine species and porcine species. The canine species IPS cells we're developing both for therapeutics and drug screening, as well as in the future for gene editing projects. But this morning I'm going to focus on our cellular agriculture portfolio. Next slide, please. Why cellular agriculture? Well, cultured meat is now seen to be part of the solution for future food security. Animal-derived stem cells offer a key opportunity to address the protein gap by providing a nutritive biomass of cultured meat for innovative food projects, addressing in particular issues relating to animal welfare, food safety, and the impact of livestock farming on the environment. Next slide, please. there is a huge increase in the number of companies that are looking at cultured meat, and these are companies' investment and proliferation is based around a number of key advantages of the production of cultured meat over livestock production. Certainly as a sector, it won't replace livestock production, but I'll add as an adjunct to help address the protein gap. And on this slide are just some of the key targets that cellular agriculture addresses in the production of protein from livestock, particularly with respect to this session and conference, the impact of the production of protein from these animals on the environment through greenhouse gas emission, land use and water use. Next slide, please. The market itself is proliferating at a fast rate. And in the last couple of years, there's been over, I think that's probably over $600 million of investment in cultured meat companies worldwide, such that there's an ever increasing growing number of these companies in this space, totaling at least 40 at the current time. Despite the investment and the derivation of these companies, there are no commercial products currently available for cultured meat, but many products are in development derived from animal stem cells, and the development of these products is expected to grow exponentially over the next 3 to 5 years. As part of that journey, the industry does need fit for purpose tools to develop their cultured meat prototypes, and these include robust immortalized cell lines and media solutions that allow the scale up of cultured meat production cost effectively for the market. Next slide, please. Thus far, most companies are looking at the production of cultured meat from the biopsy of muscle cells from live animals, in particular bovines. These muscle cells are then amplified to produce many cells and are harvested to create a commercial product. A major disadvantage of these cell lines is that they only undergo a limited number of cycles, and so a regular supply is required from the live animals themselves. Furthermore, these cells can't generate cells of other tissue types such as fat, which you would require for the flavour required in the end cultured meat product. New slide, please. Our solutions for the cultured meat sector are based on the production of induced pluripotent stem cells, for which we currently have a patent filed. This removes the requirement for the repeated use of live animals to derive the starter cultures, and that will help to maintain quality of the product through every generation of the media. Furthermore, these cells are reduced to return to a naive state and have the potential to differentiate into any cell type. We have developed IPS cells so far from pigs and have produced a proof of concept to show that they can derive into muscle and fat as well as bone type cells. We are also at the stage in our process of looking at how we can scale up porcine IPS cell production, including in that the production of cost-effective media. Next slide, please. Our cells have been shown to be generally efficient, efficiently sourced, cloned and passaged indefinitely for at least a year over more than 60 cycles. We've shown that subcloning and serial passage it reduces the presence of the vectors that we use to induce the reprogramming, and we have characterized these in detail, including the identification and characterization of the pluripotency and transcription and surface associated markers. And as I mentioned earlier, I've demonstrated differentiation into the key cell types for cultured meat production. Next slide, please. We're now on our journey with cultured meat, and we've already got commercial customers in this line who are deriving our cells for their own products, and we're currently planning to expand our cultured meat line to different species, including cattle and sheep. We've also done some work with primordial germ cells, and shown here is our prototype chicken nugget that we produced last year, derived from avian PGCs for the production of cultured chicken. Next slide, please. I was quite a cynic, having come from a veterinary and agricultural background as to cultured meat, and I've really been on a very steep and exciting journey with Roslin Technologies in this space. And when you start to look deep, you can see this is a really vibrant space, and the reality of cultured meat is coming soon to us. Most notably, the regulatory approval in Singapore of lab-grown meat for a chicken product in the last month. And the significant amount of funding that's just recently gone in as well to cultured meat from seafood. There's been quite a lot of if you Google, you'll see a lot of studies about how people feel about this and the uptake potentially of cultured meat in the future across different generations. And if we can make it cheap enough, it could even replace some components of pet food. Next slide, please. I'd just like to thank my colleagues Jo and Melvin, all the fantastic stem cell work that I presented this morning, and that's me finished.